Hello, everyone. Good morning to those in our time zones. Thanks, everyone, for joining today. It's my pleasure to introduce the first seminar of this new semester, at least in Chile. I am aware that semester times are different all around the world, but for us, it's sort of a new half a year, basically, of events in our initiative. And it's also exciting because today we have the first software tool actually in our seminar. So as some of you who've joined before already know, our initiative LibreHub is a CZI-funded initiative to help spread awareness around the possibilities to build your own open source hardware to use for laboratory experiments around bioimaging and sample handling. And we collect all our videos on our YouTube channel. So there it's possible to see the videos from the past. A lot of people make use of this. And in fact, we now decided we will also improve even the accessibility of the videos by jumping to certain points into the videos and having little interactive menus because it's a resource that seems to be used by the community. And to drive hardware and to analyze the data you generate by hardware, you also need accessible software tools. So they're an important part of the mix. And so we're going to also have a seminar soon on the Napari tool. And in that context and the Python ecosystem, it's my pleasure to introduce Dominik Kutra, who will be talking about the Ilastik tool. And I think he can explain a lot better what this is about and why he's working on it. So I just want to hand over to you right away. And I'm excited to hear and to discuss with all of you.
Okay, thank you very much, Toby. Also, thank you for giving me or us the opportunity to present here in this community. And let me share my screen really quick. Okay, can you all see my screen?
Yes.
Very nice. Cool. So, yeah, welcome also from my side. I work as a software developer in Anna Kreshuk's lab at the EMBL in Heidelberg. I work with her like for 5 years now. And what we are developing in this lab is a software tool that is called Ilastik, which claims to be a user-friendly machine learning-based image analysis tool. And of course, this tool is open source. I'm a little bit of an open source enthusiast. And yeah, that's why I'm doubly happy to be presenting in this forum where other open source enthusiasts are also. Okay, and I've made a habit when I start like any presentation these days, the first thing I show is this little graphic. So this is what you see if you go to this website, forum.image.sc. And these are all the image analysis tools from the open source community. Basically, they come together in one joint forum and everyone is answering questions. So this is our main way to interact with users. So you can go there if you have questions for a specific tool, for example, Ilastik, but you can also go there for specific questions to all the other tools, or in general, if you have image analysis questions of any form, you can go there and they are often like answered, or you get good tips where to look. Like while I was on there, I also discovered that there is this forum microlist.org. And I think this is, I don't know if you even know it, and I have also not interacted on this forum much, but on the website, if I quickly go there, oops. So, so this is also a forum in a similar manner, I think, with the same motivation to bring people together who work on microscopy hardware. And maybe this is also a good way for your community to interact a little bit there. Okay. Back. Yeah, right. So usually I say if the only takeaway is that you now know where to go and find help, this is already pretty good for my talk. But let's see if we can even like add more to this. And maybe a little bit about the group I'm working in. So this is Anna Kreshuk's group, which is here in the middle, in the center of this photo. And the cool thing about our group is that we have two parts. One of the parts does all the scientific work. So this is concerned with image analysis, algorithm development. And you can see a few examples on the right side of that. But there's also this other part, this Ilastik part with software developers. And there the idea is that once the research side has developed something interesting that we find could be applicable for other people and problems as well, then we try to make this available through Ilastik in an easy to access manner. So what is the idea? So the idea is to make machine learning based image analysis algorithms accessible to like the domain experts, so the people who are doing the actual biology, the imaging, who know what they are actually looking at. And this project is not like it's not super old, but it is fairly old, I think, for a software project. It started in 2010 in Fred lab at the University of Heidelberg. And Anna was his PhD student, and once she finished, she got more and more into the development of Ilastik. And when she got her group at the EMBL, she took the development team with her. And the central idea is you give the labels, and Ilastik solves the problem with machine learning. During this talk, I will also give a few like live demos on how this looks in practice, and this will be the first one. So I'm going to start Ilastik. And the first step, of course, is to load an image. I've prepared some sample data, and this is, I don't know, one of the data sets I show all the time. There we have two different types of cells versus some background. And the task here would be that we want to separate the cells from the background. And for this in Ilastik, after loading the image, you go to the next step where you have to select some features. And don't worry, I will explain later what these actually do and what these are. But for now, just bear with me. So after selecting some features, you can go right to the training step. And this is where, in principle, the fun begins. You decide what to do. I want to select segment cells versus the background with two labels. And then you start like drawing on the image. You go to live update. What happens in the background is a classifier, a machine learning classifier is trained based on the labels you have given. And you get instant feedback more or less. And then the idea is that you go on and correct the classifier where it's wrong until it's good. And here in this image, you can see that the yellow of the cells, it goes out into the background. So this is not good. There we want to add some more background annotations for sure. But then, of course, the cells get less well recognized. And sort of in this interactive approach, you go on and on and on until you are happy with the result. And you see the feedback is quite fast. And you can do this basically on images of any kind. So they can be 2D, 3D with multiple channels, and also time series. And that's it. And of course, you cannot you are not limited to two different labels or to a single image. You can train a joint classifier with multiple images and train something that generalizes well over new images as well. Then in the end. Okay. Saving this. Let's maybe go on. Maybe about the people who are developing Ilastik. So currently, this is the development team. We have Anna Kreshuk, the group leader, and then we are 4 developers. But in the group also we have some software enthusiastic pre-docs. Adrian Finn, who also contributes now and then to Ilastik, which is really cool. But since it's very old, you can probably imagine that there are many people who should be mentioned here who contributed to Ilastik, and I will highlight 2 here. Fred Hamprecht, who hosted the development team and supported it for a long time at the university, and Stuart Berg, the previous core developer of Ilastik. And well, of course, we would not do this if we wouldn't have any users. So we interact quite happily with our users. Usually they find Ilastik very useful. And of course, they find ways to improve it as well. And for this, we are really happy for this feedback. We are happy to be in the bioimage community because it's such a nice one. Very cool people there, very friendly environment. We also rely heavily on the open source community. Ilastik as a software has 250 or even more dependencies itself. So that's a lot of work of other people that we rely on. And of course, we are thankful for having received funding or still receiving funding from various sources over the time. Okay. But if we go back, so if you remember what I showed you in practice, so roughly what I've done there when I loaded this image, I went on to select some features. And what are these features? In Ilastik, those are generic image filters. What they do is they take the image, they perform some operation on this image that highlights, like produces a new image that highlights certain characteristics of the structures that you have in the image. For example, in the center, you can see something that highlights boundaries, or on the right, something that like highlights certain textures. And after having selected those, you, as a user, you add some training data, and this I've done by painting on the image and giving examples to the algorithm, what I like expect in these two different classes. And then the machine learning algorithm does like takes this training data, looks at these various image filters, which it uses to characterize every pixel in this image, and comes up with a prediction for the whole image. So that's in a nutshell what you have seen before. And I will try to like go into a little bit more detail now with this central part, this machine learning algorithm part. So what is it doing? Why does it work? And like I will do this with this example. This is pretty zoomed in EM brain data set. Like what I want to highlight first before I start is that, I mean, probably you all know this, but this is important to realize that this image, we look at it as various grayscale levels, but what is saved on the computer is a sequence of numbers. And this is why this all works, right? The computer, we just choose to look at this as grayscale values because it's like pleasing to the eye and easier to see things, but for the computers, it's all numbers. So, and when we then start annotating on this image, like for example here, I annotated some boundary pixels in blue and some of the cytosol or of the like cell body pixels I highlighted in orange. And what we can do now is look at these numbers. And we can see, so I coded them here as gray level, but they are like a continuous numbers as you've seen before. So we can look at those at on an axis. And you can see that there's a tendency that, for example, these orange pixels, like these pixels that I've marked with orange, they tend to have higher values, while the blue ones tend to have lower values. And this could give you the idea, maybe I can separate those, right? But if we add some more annotations, we see, okay, there's quite the overlap. It will be really hard by just like taking one number along this grayscale, gray level axis to say they belong to the one or the other class. So what is done in Ilastik is we look at not only one value, we look at multiple values. And I have like one that is actually not used in Ilastik here, but it is just for the example, which would be the mean gray value in the neighborhood. So what you would do is here, you compute a second feature, you go to each of the pixels that you have marked, and then you go into the pixel neighborhood, you look at all these values and you compute in the mean of this. So this is a new number. And then you can look at those like also on an axis. And again, you see, there's again an overlap. This number will also not help. But if you go then and interpret these two numbers as coordinates in a space, the feature space, you can see that, okay, maybe here, these two classes look separable. And until now, we haven't even done any machine learning, right? We have only thought about features that we can use to characterize certain pixels. And when the machine learning comes in, this is actually like now, it takes this training data and it computes some sort of separation between this training data in order to put this into the two respective classes. And the cool thing now about this is once you have this this line separating those two features, you can now go to all the other pixels in this image and do the same kind of feature transformation. So you look at the gray level, you look at the mean gray level in the neighborhood, and then you only look at what side of this line these come out, and you can assign a meaningful label to them. So this really is like oversimplified how this works. And practice, these feature spaces, they look not that easy separable, and also they are in general of a higher dimensionality. So for Ilastik, it's usually like 40 or more dimensions. And then, of course, you need something more complex, like a line separating the two classes. But this, like in principle, is exactly how it works. Yeah. So, but so far I've like touched upon one of the workflows in Ilastik. So this is the startup screen that you see when you start up Ilastik without specifying anything. And you can see we can do various things here. We have, like what we have seen and what we have talked about so far is pixel classification. And what I want to do in the next minutes a little bit would be to go over most of the other workflows and give you a feeling for what they are doing. Okay, for completeness, I will also show pixel classification. So the idea is you have some sort of image. Again, it can be 2D, 3D, time series with multiple channels. Everything is allowed there. So up to 5 dimensions, and you are interested in finding, like assigning a semantic, meaningful label to each of the pixels in your image. And what you have to do for this, the only thing you have to provide, as you have seen, you have to add some labels for the like give examples for the different classes, exactly how I showed before. And if you look at this image, this works okay, but not perfect. So there is like basically a next level of the same approach, which we call the auto context workflow. There you are again interested. You have some sort of input image, and you are interested in semantically labeling all the pixels in this image. And what you have to supply is you do pixel class, like exactly like in pixel classification. You do it once and like mark all the different things you have in your image with different classes. And then the trick is that you don't stop here, but you take the output of this like first classifier that you train, and you feed it into a next, like into the second stage, and you do pixel classification basically again, but not only on the raw data, but also on the predictions for all these classes you have added before. And what we have found with this is that you get a much better result, but this cost, of course, comes also at some cost, because you have to compute much more usually. So if you, for example, want to do this in 3D, I would say you shouldn't even start without having 64 gigabytes of RAM. Yeah. But looking at this image, this is still not perfect, right? You can see like there are pixels here and there that should not be there in a clean result. So what do we do about this? I mean, first of all, how well does it work? This combination that you've seen, these generic features with the nonlinear classifier, is a quite powerful combination. Before deep learning, I'm sure you have heard about this, it was actually state of the art. So this was the best you can get. And it still is around because it can solve many problems, especially with biomedical imaging quite well. And it works with very little training data, as you've seen. You can live train something that will give you a result. It's very fast. But what you have to keep in mind is that these generic filters, this is the last point here on this list, they are local. And what do I mean by this is that like if we look again at this some EM volume, or one slice of it, and if we zoom into this volume, you can see here, I mean, you have to take my word for it. I also only know this because people have told me this, but this is the mitochondrion here. And you can see this because it has like some squiggly stuff here. So as a human, you can, like if you zoom in like this, you can see clearly this is the mitochondrion, easy. But if you actually zoom into a state where Ilastik, actually, this is the context that Ilastik sees for each pixel roughly. And there, even as a human, if you would want to classify this boundary as being like a real cell boundary or a boundary of the mitochondrion, you would like not be able to do it, right? Because you cannot even like see the right information. It's not there. So this is often where Ilastik then fails. That is what you have to keep in mind if you need like long range information, and this kind of pixel classification approach might not cut it. So what are we doing about this? Our group, I mentioned before, has these two parts, this research part and the software development part. And in the research part, like there have been like beautiful segmentations of like whole animals, like for example this Platineris segmentation done by Konstantin in our lab, and then there's also the like work on various plants done by Adrian in our group. And yeah, just what has emerged there as a very good pipeline for this kind of dense segmentation is this approach where you have some sort of inputs, you train a neural network to get boundary predictions, and then you do some more or less smart graph partitioning that gives you a final result of the dense segmentation for like the whole image. And of course, like we want to make this available for everyone to use this kind of approach, but it's actually not that easy. So if we look back at what I've shown before, like this kind of pixel classification approach with the generic filters, where you had like this little bit of training data, right? You had what we call sparse annotations. You only annotate a little bit throughout the whole image, and then you were done basically. But what in the deep learning regime, first of all, you say you don't even select any filters. You don't do that anymore. But what you offset this with is adding a lot more training data. So usually what you need is really many images densely labeled. This means every pixel actually hand labeled to the respective class. But then, of course, you are still not done. So training a CNN from scratch, a convolutional neural network, basically the machine learning approach that is currently used for image segmentation, means this dense labeling training data, but then you have to choose a framework, choose an architecture. So what kind of network do you want to use? You have to configure hyperparameters. So those are numbers that you cannot really train. And then, of course, you have to write data loaders, augmentation routines. You need a machine with GPU, and you need a lot of time. So this still takes a PhD student a couple of weeks or months, depending on the complexity of the problem. If this has to be done from scratch, especially like this dense labeling of training data is a huge bottleneck. And in order to overcome this, like there's a community initiative, which is called the Bioimage.io Model Zoo. And there the idea is that like to make these networks that like so much work went into to make those available to everyone to run on their own data. And this is where this Bioimage.io Model Zoo idea was born. In practice, what you do is you have some sort of image. What you are interested in is, for example, a really good boundary prediction, like here. And what you have to do is you go to the Bioimage.io website, you search for a model that suits your needs, and then you run it. So this is like in development for quite a while now, but a preprint is already out. And maybe I will show you now in practice how this would work. So repeat demo 2. Okay, so I've already loaded an image and proceeded to the neural network running step. So the idea is you go somewhere to this Bioimage.io website. Let me maybe do this. And you are looking for something. So our image is 2D and it's some sort of plant. And then you see there's 2 networks that we can choose from. And how to apply this network now is you can either copy the Zenodo ID or like the funny animal name, the nickname. You go to Ilastik, you paste the name here, say click on this button, then the network is downloaded, it's initialized, and you're ready to apply this. So this on my machine, this runs on the CPU, so it takes a little bit of time, but you can see already that some predictions are coming in. And this is basically a very, very easy way to apply neural networks on your own data. But of course, this comes with all the dangers that you can imagine with neural networks. So you have to really make sure that these actually work on your data. You have to look at the result and make sure that it doesn't yeah, fantasize something or highlight something that is not actually there. Okay. Yeah, and the second step, this graph partitioning step, was already in Ilastik. So this is what we call the multi-cut workflow. And what this does is, I mean, you are coming in with an image with objects that are surrounded, separated by a boundary, and you are interested in the dense segmentation of this. And what you have to provide is, as a user, you add training data in order to recognize boundaries that should be kept and boundaries that should be thrown away in the final result. And this I also wanted to show in practice really quick. This is again this image that you've seen before. And in the multi-cut workflow, you are not done by loading a single image. You have to also load a second image that corresponds to this first image. And this is actually the image that I've exported from the neural network workflow that you have seen before with this network. So this is you need a probability map for the boundaries for this workflow. And then in the next step, based on this boundary probability map, you compute the watershed of the distance transform. And this is in order to make the problem a little bit more easy to solve. Because in the end, what you are trying to do is you want to assign pixels to form objects. And this is, of course, there are many pixels even in such a small image like this. But to make this problem easier, we first have this step of the watershed that groups pixels together, like with an approach that is like not very smart, but it groups the pixels that can confidently be merged into larger chunks together very well. So you can see, for example, this here, like is probably a single object, but you can see it is in multiple parts, but those parts were very easy to merge. And then with this, once you have done this, you can go to the next step, to the multi-cut step, where you can either, like if you're really, really confident in your probability maps that you have supplied in the beginning, that they are really, really good, you can try to solve it immediately. So if you click on this update now, it only looks at the probability at the boundaries that you have supplied as input data and tries to solve this problem of merging the pieces together. And as you can see, it does so quite okay, but it doesn't get everything. For example, here, this boundary is not in the solution. So all the blue ones, excuse me, all the blue boundaries are the true boundaries. And all the yellow boundaries that you can still see here are the boundaries that are thrown away.
Just to clarify, so you have to hand mark these boundaries, right? So here you went through and you told the algorithm that the yellow ones are not true.
Sorry. No, here actually I didn't do that yet, because I was very confident in my boundaries, and it would only look at the boundaries that I've supplied in the beginning. Like it would look along these edges and see how good the evidence actually is. So it would basically look at this data along the boundaries that are still there, and you can. I don't know if we have a good.
Also, the automatic update that you ran already did this classification in yellow and blue automatically.
So in yellow, those are only the boundaries that are here from the superpixels. So basically, maybe you can see this here. Here we have multiple superpixels, and if I go to the same position in this image, you can see that the yellow actually only shows the boundaries of these superpixels.
Yeah.
And now if I want to, so if I click on update now, it really only goes along all the boundaries, and it looks into the probabilities image and looks how bright it is, basically. Then like this gives it a hint on whether to include something in the solution or not. But of course, this most of the time this doesn't work, and you can also see here that it doesn't really work well. For example, here is an error, and yeah, probably here these are actual true boundaries as well. So what you can do there is you train an edge classifier, And you do this by clicking on edges. For example, here, red edges will be kept in the solution and green edges will be thrown out of the solution. And if I do this for a few, especially those that I want to have included in the solution, and if I go to live update, I first get like a first prediction of which boundaries are voted in and which boundaries would be like rather voted out. So I correct this. And if I now go to this update now button, to this multicut solution, then it doesn't look into the boundary channel. It looks at these predictions for the different edges and includes those in the cost function to solve the multicut problem. And you can see, for example, I think before this, this boundary was not included in the solution, this object, and actually now it is. So this is how you really quickly get to a dense segmentation of your whole volume. Maybe okay, I will correct this as well. No. Okay, no. Good. Yes. So that's for the multicut. I will close this. And now I will go through some like other workflows. So this is more or less like the main line for segmentation-based approaches you've seen. And now I want to highlight a few other ones that I find interesting. So the first one would be carving. This is a semi-automatic workflow to segment objects one by one sorry, in an image. And what you have to do there is you have to like indicate what is object, what is background. Carving, I think it's always better to see it in practice than to talk a lot about it. So I will show this as well. This will be demo 4. So I've already loaded some data that I want to work on. It's again some EM 3D data. And what like the main way to interact with this workflow is to give examples. So for example, I want to now segment this object here. I say this is object and the rest here around here is background. Then I click on segment. And this then gives me a first candidate segmentation in 3D. You can also see some 3D tree view here on the side. And the idea, as more or less always with Ilastik, is you you correct it where it's wrong until you are satisfied. And after seeing maybe the multicut workflow, you might ask, why is this even interesting? Because you can do this all in one go for all of the objects in your image. But sometimes you are not interested in all of the objects in your image, but only in a few of them. And you can do this, right? You can say, for example, save it here and go to the next object and segment it like right after. And maybe you are only interested in seeing how these two objects relate in 3D, for example. So you can show this in 3D also, and you see, okay, they are very close together and hug each other in a way. I don't know. But you can do these things with this workflow very efficiently and very quick. So, and especially if you're interested in generating, let's say, ground truth data that is very accurate, because of the nature of this workflow, as you have to go and correct the segmentation until you feel it's really correct. And so you do this one by one, you will arrive at a fairly good segmentation in the end. Okay. Then, I mean, until now, everything has been more or less pixel-based, but we also have workflows to deal with like object-based problems. And this would be object classification. What is an object in Ilastik? In Ilastik, an object is some like pixels that belong together. This is basically the unit now. And as opposed to pixels, where you would reason in terms of their neighborhood, like you can look at the pixels right in their vicinity. With objects, you can describe them in way more powerful ways. For example, an object, like it starts off, it has a size, how many pixels is it, or it has shape characteristics. You can analyze the intensity characteristics. And in the end, you are, for example, as here with this image, you would be interested in characterizing two different types of cells. We have the bright ones and the darker ones. And you can do this in a fairly similar manner as you have seen with pixel classification, but this is not the only thing you can get out of this workflow. You do this, of course, by clicking on examples and training a classifier. But this is not the only thing you can get from this workflow. The maybe more important thing you really get out of this workflow is a big table that describes all of your objects with many characteristics. For example, it's characterized the shape a little bit. It gives you gray level characteristics, intensity characteristics of your objects. And with this, you can basically go on and create plots and get more insights into the data you are looking at. And let me look at the time. Maybe I can show this really quickly as well. So I've already preloaded some data. In order for object classification to work, you have to supply it with a second image. This one I created in pixel classification. So this is basically the one from the first example, where I did foreground, background segmentation. And this helps me now in this next step to go from pixel probabilities to objects. And this step is called thresholding. Here we work on the boundary on the probability map for like cells versus background. Everything here is selected correctly already. If I hit apply, you can maybe see what I mean by objects. So now everything that has a different color is in fact a different object. So this is the first step you have to do. And once you have done this, you have to select some features. I'll do this really quickly and jump over this. And you have the binary image. Then you can classify it here, for example, bright versus normal cells. Sorry. So bad. Okay, and you do this by clicking on the cells. You also click some normal ones. You go to live update immediately. And then you correct it where it's wrong. So for example, this one, no, this is correct. Let me find one error at least. I mean, what we can do now is we can add a label, for example, add segmentations, because I see some, and label these cells that are merged together, or these little things that are not really cells. Go to live update again, and you can see, oh, now it has recognized a lot of them as actually being bad segmentation, so we have to correct this. Yeah, and with this, you can see bright cell that you can get like interactively again as before. You can get to a really nice result where it separates two different types of cells, and even the ones that you maybe want to exclude from your analysis because they were not segmented nicely in a automatic fashion. Okay. And final workflow, close to the final workflow that I want to show. This is tracking, and in tracking, you are interested in how something in your image evolves over time. So this could be cells, for example, or nuclei, as you can see here. And here we are interested in how maybe they move in the image, maybe they divide. And if you want to reconstruct this like fate of a cell over time or of any object, then you can go to tracking. And so that in the end, you know that, for example, this cell moved here, and this cell divided into these two, and so on, like this kind of information. And what you have to do, and this is very similar to what you've seen just in the last demonstration. You have to train two object classification classifiers, one to recognize when your objects divide. And if you don't, if your objects don't divide, for example, if you are tracking mice or something like this, then you don't have to do it at all. And the second classifier you have to train is an object count classifier, because sometimes, I mean, segmentation will never be perfect, right? You will always have objects touching each other in a way that you cannot separate them with the techniques presented here. And in order to deal with this, still through the tracking timeline, you have to teach a classifier how to recognize objects that are more than one. So that in the end, you can still see that two objects, for example, they started separate, they came together, formed a single one, and then they went separate ways again. So this is why you have to train an object count classifier. And in the end, you get not only an image with your result, but also a huge table which describes the fate of each cell over time in a lot of detail. All right. So this is basically what we have currently. And yeah, I think I will take 5 more minutes to show you what's coming next. And this goes along this neural network problem that they sometimes generalize not very well, or you have to train them really, really, really well in order for them to generalize well. And so this means that you can take any neural network and apply it on your own data that was never seen in training or something like this. Maybe I can, so the next thing that I will show is called trainable domain adaptation. And I here have examples of, again, electron microscopy. And you can see here mitochondria from different data sets. So these are the dark thingies in the image. And if I do pixel classification, so doing this with pixel classification is really, really hard for some reason, because they look so complex, everything looks kind of the same, and there are many other things in the images usually that look very similar. So in general, you will get results that are kind of like this. I mean, you can get a little better, but I wanted to show that, yeah, often you cannot. So you can see everything that is yellow is supposed to be mitochondrion, but for example, here in the middle data set, there are lots of boundaries that are still highlighted in yellow. And here, yeah, on the right also, there's some smaller stuff. But as you can see, it's like more or less really, really hard. And so someone in our lab had the idea that actually, that you can maybe train a convolutional neural network as before to correct the predictions from the random forest rather than doing this from scratch from the original image. And so the idea here would be that, I mean, mitochondria, they look the same more or less, like in terms of shape on many data sets, right? But the characteristics of those data sets are fairly dissimilar. So this is what we call a domain shift. So going from one image to the next, like there's a huge gap because the characteristics are so different. But if you look at the random forest predictions, they have fairly controlled characteristics. So they, like, the value range is always the same, and also maybe the mistakes they make is also the same. So the idea is here really, so this would be like how you would normally train such a convolutional neural network. You would take the raw data and you take dense annotations. So again, every pixel or voxel in your data set is annotated, and you take this as training for many data sets. You train for a while, and then you get your cool convolutional neural network that will segment data very, very well. But what was proposed in this trainable domain adaptation work was actually that the convolutional neural network will never see any of the real raw data. So what it only will see in training and also in prediction time is these dense predictions that come out of pixel classification, so out of the Ilastik pipeline. And of course, this is automated. So we take the dense annotations, we sample in an intelligent way, we generate these kind of Ilastik-like results, and those go then into the training. And again, maybe seeing it is better than talking about it. So this will also be, I think, a very quick one. So I already loaded some data in the beginning. As pixel classification is involved, it looks all the same. I select some features, and I will start in the next step to annotate the things that I'm interested in. And this would be mitos in this case. So I start here, add some background labels, and go to live update immediately. And what you can see is, the result is not very good. So I corrected more. Then other stuff from the background comes up, and maybe you get a feeling that this is actually not that trivial, not as trivial as with the cells you've seen before. So it's, not easy. And again, in this trainable domain adaptation kind of domain, you would now actually not feed the raw data to the neural network, but some dense predictions, and the dense predictions we just generated with our random forest here. And the only thing now we have to do is to choose a suitable network. These networks are also on the bioimage by our model Zoo, and are automatically populated here in this workflow. The ones we have. I should take the right one. You initialize, you download the model from the website, initialize it. And then the only thing that's left here is to say on which channel it should operate. This would be the Mito channel. And then, so what you see now is the predictions, the neural network predictions that come out of by looking only at the, basically only at this yellow data that you can see here. So it takes the yellow, it puts it into the neural network, and it corrects it. And of course, this is still not perfect, but the idea now is that you go on training your random forest. You make this better, and so this regenerates new predictions and does the neural network prediction on top of it again. Like this, and this over the time. So basically, you are assisted by a deep neural net to come to nice segmentations. And once you're done, you can do this for the whole data set, or for multiple data sets afterwards as well. Okay. I think that's all about this.
Hey, Dominik, just a heads up that in about 5 minutes, the hour is full. I hope that we can also discuss a little bit, a couple of more questions. I don't know, some people might have to go on the hour. I can stay a little bit longer. And the video recording but just that you have an idea of the time. So I guess if you would be able to wrap up, and I don't know.
I can wrap up maybe in 1 minute. So the only thing I want here as a maybe as a takeaway is that also, like, you're not done. Once you have trained something, you're not done. You can do more things afterwards. You can run your classifiers that you've trained on your data. You can run those on new data that they will not include in the training sets. You can run this in various ways, do this in various ways. For example, there's a Python API for pixel classification. This is all the code you need to predict with a pre-trained project. Then there's also a batch processing UI in Ilastik itself, so you can drag and drop as many images as you want and process them with your trained classifier. There's the headless mode for more more control over this whole process. And also, if you want to run on servers that don't have a graphical user interface. There's a plugin for Fiji that is really, most importantly, it allows you to import and export results in our preferred format, HDF5. Everything is macro recordable, and you can also run pre-trained workflows on there. So yeah, and it is really, so Ilastik is open source. We provide binaries for all the three big operating systems for Linux, OS X, and for Windows. You can go to our website, ilastik.org, and download it. If you have questions, go to the forum. We are happy to talk. You can follow Anna on Twitter. And even like if you're really into it, you can contribute on our GitHub, of course. And with that, I would be happy to go into some questions.
Thank you so much, Dominik. It was really fascinating. It's even way cooler than I thought. So I have a lot of questions, but I'm sure some other people might have too. This is the advantage of joining us in the live session. I know that many people watch them afterwards, but only here you get the convenient opportunity to ask direct questions. Anything was unclear in terms of content or language for anyone, or you have a follow-up question? Please go ahead. Okay, Maria.
Hello, thank you, Dominik and Tobias, very much for this possibility of knowing, because I didn't know about before about this software. You showed us grayscale images, and I was thinking that when working with, for example, double labeling immunohistochemistry, when you work in overlay images with the two channels, We need to go to the color world in some way, or we may work in parallel in grayscale images of one channel and the other. So what is the more what do you recommend for Ilastik to work when you are trying to, let's say, for example, identify double labeling, immunohistochemistry at nuclear and cytoplasm of the same cell. So there are many tasks that can be interesting to know how do you suggest to work. Thank you very much.
Sure. So, Maria, I'm not an expert in all these particular tasks, but what I can tell you is that you can put in as many channels as you want in Ilastik. The only thing is, Ilastik is not particularly great at displaying multiple channels. So it will try to do something. So it will give you colors for the first 3, but if you add more than 3 channels, then it will show you grayscale images of the individual channels. But what is important, everything you give to Ilastik, so all the channels go into the analysis. So the features are computed in all the channels. And if you have some, for example, some channels that should overlap in certain positions, and you want to find those, and this is absolutely possible because it will learn it.
Okay, so you can run in parallel each channel and then see if there is overlapping in the positive nuclei in one channel and in the other, for example. Okay,
exactly.
Okay, thank you very much. I'm sorry that I have to leave and I have another meeting. And thank you so much.
Thanks for joining us. Right. There's no other hand up right now, so I'm going to jump into the question.
I have a real hand because I don't find the...
yeah, go ahead.
And I see that we have the same throat problem, actually. It's quite a pain. No, it's a very nice program. So I tried during the pandemic because I had nothing to do. It's extremely nice. And now I'm just wondering, does it work also on ARM's processors? So it will work on Raspberry. I don't care about how fast it will be, but will it work?
So you mean ARM in terms of, I don't know, like on a Raspberry Pi? Or?
Raspberry Pi, yeah.
So not yet. And I mean, so what we are doing right now is making it run on the ARM64, like that is supplied with newer MacBooks.
Yeah.
And I'm currently in the process of moving all of our dependency basically off to Conda Forge. And I think then it should be doable once we are there to also build them for ARM64 Linux.
Okay.
But this will take some time, and like I cannot promise anything basically.
No. But now they are quite good also for doing this kind of job. Okay, thank you.
Yeah, I agree, and I would also love to see it. But...
Yeah. Thanks.
This is a key question. I would like to follow up on this a little bit. So, I mean, in our context of hardware development, we often have the issue if you want to use some smarter workflows or computational imaging, or even just more data acquisition on sort of low-cost instruments. It starts to be a problem how much data you accumulate and how you handle that, and if you want to upload it with Wi-Fi or something like this. So a segmentation tool like this would be amazing, for example, to actually cut out regions of interest automatically and only save those and only upload those, for example. And I'm wondering if there would be a possibility. So most of these devices are, a Raspberry Pi driven, or maybe a Jetson Nano or something. So a little single board computer. They're not so powerful. But I'm wondering now that I've seen your presentation, if it could be possible to take, let's say, full images, segment them on a personal computer to generate this model, and then use the model on a Raspberry Pi, because there it looked like you can just use it in a normal Python script. I don't know, do you need to have full Ilastik functionality to do that? Or how does that look? Does that make sense?
Yeah, absolutely. This makes total sense. And so again, but like Ilastik is like the classifier you save in this project file. This will only work if you do the full pipeline. So if you have the same filters, and use the same random forest implementation, and then you get like results that would be, I say, are reliably the same. And there, there's this little missing link that Vittorio also asked about, like, can we run on ARM64? Because, for example, our filters would have to run on there. The other libraries that are involved in this whole processing would also have to run on there. Not all of them, as in the UI that we've seen, but I mean, there is an overlap, of course, and not all of them are available yet as ARM64 builds. So, I mean, I also would really like to see it, I have to say.
That'd be really great. I don't know, what kind of processors in the Jetson Nano? Do you know of it?
I don't know.
Still an ARM.
Yeah.
Still an ARM.
Okay.
Yeah.
Yeah.
Okay, but yeah, that would be absolutely awesome. I think we're your first customers of open source, open source. But anyways, first users.
I mean, if you really are interested in, then I would invite you to open an issue on our issue tracker about this, because if it's only me that is saying that we need it, then I don't know if Anna is so convinced, but if there's more people interested, then yes.
That's a great idea. I'll ask everyone in my team to open and support me. That sounds great. So I have another question. If do you have to run off, or you still have a few minutes for question? I
have time.
Okay. So I'm wondering about the machine learning incorporation. So apologies for my ignorance. I'm not entirely sure the definition of machine learning, sort of where it starts and where it ends. So where do you have, I don't know, a clustering algorithm, and when can you call it machine learning or so? But I understand that there's certain types of models that you can generate with Ilastik, but you can import them, right, from this bioimage IO, if I understood this correctly. So
yeah, so everything you generate currently in Ilastik itself will be what we call shallow machine learning. So this is a classifier that is trained with very little training data, that is very fast to train. So these kind of models, so the classifier we use is the random forest, and those models you can produce and you can save and you can load again. So this is one part, and the other part with these more involved deep learning models, it is as you have said, yeah, you can load them directly from the bioimage IO model Zoom, the ones that are there, and you can try them out on your own data. But
right. And I guess you've tried a couple of those. How well does that work? Because I always had the impression that it matters quite a lot how you record your images. So if you have a slightly different, microscope and setting, you might get different kinds of data, and then it might not work so well. So that you need sort of training sets that are done with the same hardware, for example. And how well does it work to use these models from other people?
Yeah, I think this is like hard to answer as a general question, because there is so much that goes into it. It's not only like how different these domains of these two different images are that you want to supply. It's also how well was it trained, because you can do quite a lot with augmentation routines. So you slightly perturb your training data so it looks a little bit different. And if you do it in a very intelligent way, then you can actually generate a model that is like generalizes to some respect, but it's really hard to tell in advance. So right now the advice would be to try it out, because it's like this is what our energy went in so far, to make it easy to try out, so you at least get a quick answer to this. And I've also seen it like not work in some instances, or not work at all. But yeah, and in the future, we of course hope that we can find better tools to this. So maybe like automatically finding the best network that is up there. Yeah.
But I guess the good thing is that in Ilastik, you can already fix them up, right? You can import the model, see if it helps, and then manually sort of define some things to improve it on your images, I guess.
I mean, this not really, right? So with the neural networks, with the normal ones, you are basically done. Once you download it, there's no correcting them, because retraining or fine-tuning such a neural network is I mean, you need like it's not enough to just supply a few brush strokes. But this other,
You
like the
don't
last
combine
example,
it with the random forest model afterwards. Okay, I see. So it's one or the
so
other.
this was, yeah. So I mean, this was the last example that I showed. This trainable domain adaptation that's like will be in the next version of Ilastik. This is not out yet, but for this is actually exactly geared towards this use case where you like have maybe a task that is fairly similar but completely different hardware, and where it's basically impossible to do this in training without having the actual data. And there, shifting this to the random forest domain where everything looks kind of the same helps a lot. But these are specialized networks that are only trained on like random forest predictions. They are not trained on normal data. So this is why it works.
Okay, that makes it more clear. Thanks. That's really great. Well, I don't want to ask any more of your voice. Thank you so much for presenting anyways today. It's super interesting. We'll upload it soon on the YouTube and put it on the social media as well. And yeah, I guess you also pointed us towards the right web pages and fora to ask follow-up questions. And I'll make sure not to forget to raise the issue, because that would be really cool to have this available for ARM processors.
Awesome. Yeah, great. And then thank you very much again for this opportunity. This was really fun. And yeah, I hope to interact a lot more with like PeopleHub users in the future.
I hope so too. Thanks again.
Thank
Bye,
you.
Dominik.
Bye.
Bye, everybody else.